Welcome to the world of Primin B. In this episode, we will explore the formation of Primin B and how its first life evolved. Primin B will be one of many planets that exist in one story about humanity discovering a vast universe that is teeming with life. Primin B's story began 12.5 billion years ago with a massive molecular cloud in space. Over time, this cloud gained mass until it was so massive that it collapsed under its own gravity. As this molecular cloud collapsed, it became increasingly hot. Eventually, this heat formed a star. Remaining matter left over from the star formed to make five planets in its orbit. The second planet from the star, Primin B, falls within the star's habitable zone. Its single moon was made elsewhere in the solar system, but got trapped within its gravitational pull. Primin B is a rocky planet that is slightly larger than Earth. This planet has a molten core that allows it to be tectonically active. Gases that are released by this tectonic activity will make up the majority of Primin B's early atmosphere. More gases and other chemicals will be introduced by a bombardment of meteor impacts that Primin B has experienced. This ancient atmosphere will largely consist of gases such as hydrogen sulfide, carbon dioxide, water vapor, ammonia, and methane. Eventually, this planet's atmosphere cooled enough so that the water vapor could condense into vast oceans. Random chemical reactions between the ocean and atmosphere occasionally produced simple amino acids such as glycine. Two amino acids are able to bond through a method called dehydration synthesis. In this process, a water molecule is released, allowing a chemical bond to be created between the two amino acids. Reactions like this can continue to happen until the bonded amino acids form long chains called polypeptides. Some of these randomly assembled polypeptides had catalytic properties that allowed them to facilitate chemical reactions. Some catalytic polypeptides are capable of self replication. These self replicating polypeptides eventually created proteins that could create self replicating RNA. Since this RNA could self replicate, it was subject to natural selection. Errors in its replication may change its chances at replicating more. One clade of RNA began to synthesize and coat itself in fatty acids. These membrane bound strands of RNA are called protocells. Over time, these protocells became more complex. They developed better membranes, the ability to metabolize chemicals to produce energy, and eventually switched to using DNA as their primary information containing molecules. The first clade of these DNA bearing cells is what we will consider the last universal common ancestor, which will be abbreviated as LUCA. It is a heterotrophic prokaryote that metabolizes nutrients dissolved in the water. From this one organism, all of Primin B's life, and life across the Milky Way, will descend. This first life appeared 9 billion years ago. Around 8.8 billion years ago, one clade of Luca was isolated around hydrothermal vents at the bottom of Primin B's oceans. These vents release large amounts of sulfides and other nutrients into the ocean. This clade of Luca capitalized on it by developing new metabolic pathways. They began to use sulfides, carbon dioxide, and water to produce simple sugars that they use for energy. This first autotrophic clade of chemosynthesizers will be called chemophagous. Just as chemophagous was isolated to the hydrothermal vents, a population of it lost access to them and their specific nutrients. In order to produce energy, this population adapted to use the sunlight as a source of energy for their sugar producing reactions. This clade started producing orange pigments that allow it to absorb the shorter wavelengths from Primin B's star. This clade also stopped requiring sulfides to produce sugars, which allowed for a more efficient energy source. It took on a more cylindrical cell shape to increase its surface area while maintaining the same mass. This allowed it to collect sunlight more efficiently. This clade of photosynthesizers mostly lives floating in massive colonies on the ocean's surface, creating large orange masses visible from an aerial view. We will call this clade photophagous for their ability to eat light. They appeared 8.5 billion years ago. 8.4 billion years ago, after 600 million years of evolution, Luca's descendants were much different than their primitive ancestors. Its metabolic pathway became much more efficient, which allowed it to have access to more sources of energy. One of these new energy sources was other cells. This clade is extremely broad and could potentially have millions of different species in it. They will be called microtrophs for their ability to consume almost anything smaller than them. The introduction of microtrophs will mean that they outcompete Luca, making the very first major extinct organism in Primin B's history. 7.8 billion years ago, the next leap in biological innovation occurred: eukaryotism. Like most new biological advancements, this happened by accident, in a process called endosymbiosis. This process started when one microtroph consumed a much smaller microtroph, but the smaller one didn't die. A parasitic relationship formed between these two organisms. The larger host organism gave shelter to the internal organism, while the internal organism leached off of its host's energy. Both cells continued to reproduce, which caused more host cells to exist, and inside of them, more parasites. Most of these eventually killed each other, but one survived. The surviving group invested in mutualistic symbiosis, which benefited both organisms. The host organism protected the internal microtrophs, while the internal microtrophs conducted metabolic processes and shared their energy with their hosts. The host cell then developed a membranous sac that contained its genetic material. This stops the internal microtrophs from accidentally metabolizing its host genetic material. We will classify these organisms as one organism, calling them omatosoma. 6.5 billion years ago, another endosymbiosis event occurred. This time, an omatosome began cooperating with a clade of photophagus. Like photophagus, these organisms float close to the surface of the oceans. We will call this photosynthetic clade of omatosomes Aureosoma for their orange bodies. Over time, omatosomes diversified into many unique clades. One of these clades was covered in cilia, small hair-like organelles that allow cells to control their movement. This clade is also proficient in releasing pheromones. Pheromones allow cells to establish a form of communication. Using these pheromones, this clade can form colonies with cells that share nutrients with each other. Using these cilia, these cells will latch onto each other and release a biological adhesive through their membranes that ensure colonies don't fall apart. This clade is called Tenax malia, and it appeared 5.7 billion years ago. The period of time between the appearance of Luca and Tenax malia was over 3.3 billion years. We will call this period of time the Protobion period, and its organisms will lay the foundation for Primin B's future. In the next episode, we will explore how these organisms further evolve into Primin B's first multicellular life. If you would like to talk about Primin B, or keep track of future videos, join Primin B's Discord in the description below.